Biosignaling, also called cell signaling, shows up in both the biochemistry and psychology and sociology sections of the MCAT. Every heartbeat, hormone release, and neural signal starts with one thing: cells talking to each other. Understanding how that conversation happens through receptors, messengers, and signaling pathways is key to mastering everything from hormone regulation to neurotransmission. Let's break down the high-yield parts of biosignaling and how it actually works inside your body. What is biosignaling? The cellular conversation. Biosignaling is how cells communicate to coordinate behavior and maintain homeostasis. It's the body's internal language, every signal telling another cell when to grow, divide, or react. Signaling molecules, the messengers. These are the molecules that carry the message. They can be hormones, neurotransmitters, growth factors, or cytokines. Some act locally, paracrine and autocrine, while others, like hormones and endocrine signaling, travel long distances through the bloodstream. Signal transduction, turning a signal into action. When a signal reaches the cell, it needs to be translated into an internal response. That's signal transduction. Binding to a receptor, amplifying the signal and activating pathways inside the cell. The MCAT loves to test this sequence: input, transduction, output. 4 main types of signaling: endocrine, long distance via hormones in the blood; paracrine, affects nearby cells; autocrine, a cell signals itself; juxtacrine, direct contact between cells. The key? Know which type fits the scenario. Endocrine is for systemic responses, paracrine for local coordination. Receptors, where the signal lands. Hydrophilic molecules like peptides and neurotransmitters bind to cell surface receptors. They can't cross the membrane. Hydrophobic molecules like steroid hormones bind to intracellular receptors, often directly affecting gene expression. MCAT tip: if it's lipid soluble, it acts inside the cell. Second messengers, the amplifiers. Once the receptor is activated, small molecules like cyclic AMP, cyclic GMP, inositol triphosphate, or diacylglycerol spread the message internally. They act fast and amplify the signal so one hormone molecule can trigger thousands of responses. This amplification concept shows up again and again on the MCAT. Common signal transduction pathways. The G-protein coupled receptor and receptor tyrosine kinase pathways are the big ones to know. GPCR use GTP to activate secondary messengers like cyclic AMP. RTK often trigger phosphorylation cascades that alter enzyme activity or gene expression. Remember, GPCR equals G protein plus GTP, RTK equals receptor plus kinase activity. Cellular responses, the result. Once the message gets through, the cell acts. That could mean activating enzymes, opening ion channels, changing metabolism, or turning genes on or off. Different pathways, same goal: an appropriate, coordinated cellular response. When signaling goes wrong. If biosignaling is disrupted, the effects are huge. Overactive signaling can lead to cancer. Faulty insulin signaling causes diabetes. Even neurodegenerative diseases trace back to communication breakdowns between cells. That's why understanding biosignaling isn't just academic, it's clinical, and it's MCAT gold. Biosignaling connects everything, from hormones to neurotransmitters, from local communication to full-body coordination. Once you understand how signals move through cells and trigger responses, you'll start recognizing these patterns across every system the MCAT tests. With that said, here are just a few of our student success stories. Students who mastered these concepts, boosted their scores, and got into medical school. If you're studying for the MCAT, click the link below to get started with your own prep. We help students reach a 515 plus on the MCAT, and we'd love to do the same for you. And don't forget, subscribe to our channel for more MCAT breakdowns like this, and follow us on Instagram for extra tips and resources.